It's a good time to start. So, yeah, hello everyone. So, my name is Anderson Tavares. I'm a computer vision and machine learning researcher, and I would like to present computer and vision, making computers see, interpret, and act. I will just cover just a glimpse about what uh, vision inspires computers to solve some interesting tasks. And I will focus more on the vision system side, but I will talk about some algorithms that were inspired by some process that happens in our vision. I hope this presentation will inspire you to also think about how computer vision works. And okay, let's get started. So we have a lot of development. And right now computers can see and understand the world, and that's the goal for computer vision, to make computers understand what's happening in the world here. So there are a lot of tasks to accomplish that, like image classification. I will say, yeah, here. So image classification, like providing a class to a given image. Semantic segmentation, which is trying to segment areas of the image and give labels to those areas, like what is grass, what is sky, and so on. And there's also object detection, where not only giving a label to an object, but locating where is located that object. There's also instance segmentation, which is finding the actual segmentation of the object in the image. And we have also tasks related to 3D, like 3D reconstruction based on a sequence of images, depth estimation, text recognition, localization and mapping that robots can use in order to, at the same time, localize and also provide the map of the scene. We also have tasks related to face detection, recognition, image restoration, motion analysis, augmented reality, and this is by no means an extensive list. We have a lot of different tasks related to computer vision. And I would like to focus more on the vision side. But in order to understand vision, it would be nice to understand the light itself, because we see just a fraction of the light. The light is only a fraction of this large range of electromagnetic rays. And here we have also the size, the relative size of the wavelength of the ray. And for example, the electromagnetic wave that we interpret as red has a longer wavelength than the electromagnetic wave, which we interpret as blue. Before the red part, we have infrared, and we also have ultraviolet, and with a larger frequency, we have X-rays and gamma rays. So the focus will be on this visible spectrum. We say visible spectrum, but more on on a human side, because other animals can see the world in a different spectrum, part of the spectrum. So yeah, before the Greeks thought that instead of receiving the rays in order to see, they believed that we sent rays to the object, like as if we touch the object using the rays. So this is the emission theory, and the Greeks believed in that. And now let's start with just a single photoreceptor. So there is this animal called euglena, which has a single eye spot here, which helps the euglena to move. But we have the euglena has a problem that the light, I will just see, there's that animation here. So the light can come from different directions, so it's not possible to see where the light comes from. And that's why the evolution helped to get a more sense of direction, like in this way, by curving the set of photoreceptors, we could get a better sense of direction of the light. And yeah, so that's the evolution of our eye. Yeah, in order to have just a tiny hole that allows the light to hit the retina, our photoreceptors here. So we could get a better sense of direction. So just a small part of the receptors of the retina can be hit by a given light of a given direction. Yeah, so here. And yeah, we have a bunch of more evolved components here in the eye. Like here, there is the cornea, some a membrane protecting the eye here. And there's the lens, which helps focusing the light into to a specific point. And here is the illustration of the complete eye. Just one observation here. It's not the lens that is the responsible to curve the light into a specific point of the retina. It's the cornea itself that has this the role, the function, because of the different refraction indices between the air and the liquid, which is behind the cornea, which is called the humor. And also the mathematical principle behind this projection between the 3D world to the 2D world is the pinhole model. We will talk a little bit later, this one. But it's nice to see what is the difference between the eyes and the cameras. So what happens in the eyes that happens also in the camera, and the similarities, but also the differences. Some similarities that is that the camera also has the lens and the sensor, and the eyes, we could think of the retina as a sensor itself. So the eyes also has a lens and the sensor. But one difference is that to shift the focus, the sensor has to move the lens. But in our case, in the eyes case, it changes the shape of the lens. Yeah, another difference is that in the camera, all the photoreceptors are has the same behavior or quite the same component, but what makes us capture different types of light is the filters. So we need to add filters in order to filter the light. So when we receive the white light, we could filter to get red, green, and blue. And we combine those results to get one image. But before, we need to interpolate to fill those white gaps here. But in contrast, before that, so after converting the light into electric signal, we digitize, we convert into a binary format, we store it into the disk so that the file can be loaded and we interpret as an array of numbers. But so we can do some processing, some computer vision image processing here, or just plot it as an image itself. In contrast, what happens in the eye is that we have a set of photoreceptors called cones, and they don't need filters because we have different types of cones that respond differently depending on the type of light. So usually people say that we have a red, green, and blue cones, but it's a misleading terminology because it's better to say that the cones detect short, medium, and the long wavelengths of light. Yeah, so they respond differently depending on the type of light and depending on the wavelength and the amplitude. Yeah, we also have a rod which detects the brightness of the light, and we use those rods only at night or when the place is really dark, so we don't have a sense of color here. And also the threshold for the activation for the cones are really high, so they don't work in dark conditions, in dark places. So that's why you use the rods in our eye. We have also neurons that connect the cones and rods and make a path for the signal to go to the optical nerve, and yeah, to go to the brain after. And the one important neuron is the ganglion neurons, though this with the red, yeah, illustrated as red here. I can show just some animation here. When the light hits the retina, we have this pulse, yes, going from the rods and cones to the ganglion cells and to the optical nerve. That goes to the brain here. And the distribution of cones and rods are not exactly equally distributed over the retina. So in the center, where we call the fovea, we have a large number of cones. So we have a good color resolution in the center where we are focusing at. And the distribution of rods are, yeah, in the periphery of the eye. So that's we could think of having a good resolution in the center of the eye, but not so good resolution in the periphery. So they are, it's a different experience that we have in the center and in the extremes. Yeah, we also have this blind spot because we have the optical nerves going to the brain. So we don't have any receptors here, but we don't see any kind of a black region in our image when we see, because the brain fills this gap instead with with all the the light it has in the neighborhood of this region. So, but there are some experiments that can show our blind spots, and it's really interesting to see. Yeah, and before going, looking at the brain, we can also see another difference, which is when we move the camera, we have a set of frames that can represent that movement, but the camera doesn't kind of shut down during the movement. But the eyes has a different approach. When we, the movements of the eye are called saccades. This is the fast movement between points that we are focusing, like that one, that image can show what happens with the eye, when we move to different locations in the image. And and the eye kind of shuts down during the movement. So that's why it's difficult to see the movement of the eyes when looking at the mirror. Yeah. So okay, let's continue. Yeah, so one thing that we can think of is that we have 130, I will say here, 130 million cones and rods. So if we connect all the cones and rods to the brain, we have 130 cables. We could think of that, or optical nerves. This is a lot. This will fill our eye. So we will have a big blind spot. So the eye, in fact, the eye has a compressed process going on in order to reduce the number of optical nerves and also to reduce the information which is sent to the brain in order to represent the image. So for instance, in image processing, we have this compressed version of that image, which is 240 kilobytes. And if we uncompress that, we have 14 megabytes. That's not perceptible unless we zoom in the image. We can see the differences. So if we do some calculation here for the eye, suppose we have 200 brightness levels. So it means that just 1 byte is enough to represent those levels, because 1 byte can represent 256 levels. It means that one image stores 130 megabytes, or the eye sends 130 megabytes from the eye to the brain, in this case, which is a lot. And if we consider the eye the receptors firing 30 frames per second, we get almost 4 gigabytes per second. It means every 15 seconds, a disk of 120 gigabytes being filled up. Yeah, this is really a lot. But this is isn't what happened in the eye. One way to reduce this or to compress this is the concept of receptive field. which happens in the ganglion cells, this red neuron here. What happens is that there are some regions that influences the response of the ganglion neurons positively and negatively. So suppose we have a spot of light here. I will do this animation to help. So we have a response plot at the bottom, and this blue region with the minus sign is the inhibitory region. So it will affect negatively the response of the cell. And just the center will affect positively. So if we have a spot of light over the inhibitory region, the cell will respond negatively. And if we have a spot of light on the positive side, it will respond positively in this way. So you might think of the off-center cell as an inverted version of this. And this will help us to understand what the photoreceptors and the ganglion cells actually send to the brain. Let's see. The cell, even if we don't send light to that cell, there is an activity. So it fires some signals in a specific rate. We could call it baseline rate. But if we spot, if we spot the light in all the region, so the inhibitory region and this center region will cancel out, so that it will not affect the firing rate of the cell. So it will be the same. Yeah, if you see, it's kind of the same firing rate. But if we spot if we have the light just on the center, it will excite the cells. So it will increase the firing rate. And if we illuminate only the inhibitory region, it will decrease the firing rate as well. And this doesn't depend on the brightness of the light. If we have normal conditions, if we have poor conditions, or a really bright condition, because the proportion is the same. So let's see. We can use that to find an edge. Like if we have a shadow and over the complete region of the cell, it will have a baseline rate, so it doesn't send anything special to the brain. The same case happens with the bright part. If we illuminate the whole cell, it will just send nothing to the brain. But along the edges, we have some behavior here that happens because of the inhibitory region, or if we have a lack of illumination in part of the inhibitory region. So this might indicate an edge, a possible edge. And this means that the brain only looks at edges in this case. So we could say that edges are really important to recognize objects. You can see here, so that we can see that only using edges we can recognize that it's a face here. So here, one experiment that we can take. So is there a dog here? At least I can say a dog here, even if there is no color information. I will highlight here just some, the shape of the dog. This is to show that it's important to think of the edges as a special feature that we can extract from our images when doing some computer vision processes. Yeah, so this motivated us to create also edge detectors when trying to detect objects in our images. One of them is the difference of Gaussians. It's interesting because it mimics, the output mimics what happens in our brain and what happens in the receptive field of the ganglion cells. So this is just two Gaussians. So we apply Gaussians to an image represented by the F, and we apply Gaussians. So we have two Gaussians. So the result of the Gaussians are blurring images. So the images become blurred, and we just subtract those two versions of blurred images to get the edges. And we have some kind of a white edge, white border around the edge, but it's at the side of the bright area, and we have also a dark edge at the side of the dark area here, showing that it's an edge. And this also represents or approximates the second derivative of the images. So it has a good mathematical foundation behind that. And yeah, so another experiment. So if you look at one of the intersections, one of the white intersections here, can you realize some gray spots in other intersections? I can, if I look at this spot here, I don't know if you see my cursor, but I can use some pointer here. So if I look at this spot here at this intersection, I can see some gray spots at other intersections here, even though it doesn't exist. This might be explained by these receptive fields. It's like we have more bright areas around the inhibitory region, which makes the result negative, and we interpret that as a gray area. Then if we have like a receptive field along the line, so we have less inhibitory bright areas in our inhibitory region on the receptive field. So, and this also might explain this experiment where when looking at a white spot, the other spots kind of blink like a scintillating grid in a scintillating way. So we have some black spots popping up in other spots here, but it doesn't happen when we look at the spot itself, because as we saw, the receptive fields in the center of the eye are really smaller compared to the periphery. So we have a good resolution to cancel this effect. But this might not be the whole story, because probably there are more processing, because that other image has no illusion here, scintillating grid illusion. Or at least I can't see any black spots popping up in other circles. And also, we realized that the receptive field can detect edges or changes in space, but we can also see changes in time. So if you look at this first cross here, if you fixed your eyes in this first cross for, let's say, 10 or 15 seconds at least, and so you kind of see those regions disappearing, but if after that time, if you look at that second cross, you see some after images. So like some white region at the left side and black region at the right side. This is called the after image, and it's like our ganglion cells are becoming tired or bored, because the colors are not changing over time. So the ganglion cells stop firing to the brain. So it means that we see the opposite of that color, because the response is for that specific color is decreased. So we could also have changes in time as well. So our photoreceptors and ganglion cells are really good to detect changes. So that motivated us to also think about changes in time, how to compress that, how to detect changes in time and send only information that varies over time. So we have a large area around video compression. We could have complete scenes in this iframe, but we have compressed versions of that in the B frame and the P frame here in order to decrease the size of the file. Let's see more. Yeah, there are some other effects for these receptive fields, like if we have a dark background or a light background, this also affects the perception of the circle. So we can see, or we can ask which circle is darker, if it's on the left or on the right. But in fact, they are the same intensity, but they seem to be darker at the right, and they seem to be lighter because of the neighborhood. And the receptive fields might explain this perception, this illusion. We have other illusions here, like the donut illusion. Like if we look at the center, and over time, this donut will disappear, this circle here, because of the changes over time. And we also have this illusion here at the right, where we have only with the edge, the pixels around the edge, we change from the bright or the dark, but they make us believe that the whole region is dark or bright. Like the right side is darker than the bright, but in fact it's not. But this is a kind of illusion that might be explained by the receptive field theory. And yeah, so going to the visual cortex now, looking at the brain. So if we show a colored image, what happens is that that region at the top left appears. So it means that many cells in that yellow region will be activated. But if we show a grayscale image, it means that only that area will be illuminated or activated, but not the center. So it means that the center is kind of responsible for the color information, and that outer area is responsible for the brightness. So the bottom left shows the difference in the center, in the fovea, and the bottom right shows the periphery of if we illuminate, what happens with the periphery. So there is a correspondence between the eyes, the photoreceptors in the retina, and the visual cortex, the cells in the visual cortex here. And since we saw that the ganglion cells are responsible to detect changes, we have in the brain cells responsible for the orientation. They just respond depending on the orientation of the edge. Like if we have a receptive field here, which is represented as a square, as a green square, and if we stimulate the eye by showing the blue bar here in a given orientation, if the blue bar is kind of has the same orientation of the receptive field, that specific cell will fire more. This is interesting. We have some cells that are tuned to detect orientations in the edges. And also, if we look at the neighboring cells, we see that they kind of detect similar orientations. It's an interesting arrangement of cells in our brain. So we could also do that in computer vision side by using some operators, some edge operators. One of them is Sobel operators, which is convolving the image with a filter. So in this case, it's a filter that detects vertical edges, but we can also change the filter to detect edges in a different orientation here, like here. Yes. So there's another inspiration, which is the histogram of oriented gradients. We used a lot in computer vision to find edges and detect objects and recognized objects. And yeah, this is like dividing the image into blocks and calculating the gradients and counting how many gradients we have in a given orientation. So creating histograms, small histograms of gradients. So this might describe the image in a way that we can detect edges as well and the orientations. We have another illusion based on edges, the illusory contour. So the eye kind of finds edge where it doesn't exist because of the response of the ganglion cells and the cells in the cortex. And not only us, but also other animals have this ability. So if we train an animal to look at triangles, they could respond to figures that has no triangle, but kind of resembles us as having an invisible triangle. Here, yeah. Yeah, we have another behavior in our brains. Like we have also cells that are tuned to detect not only orientations, but also the size of the object. Like there is an illusion here where we have the top left figure, but in a low contrast, located at the center. And if we fixate the eyes into one of the squares, only the top left pattern here will cancel out the figure at the center. But even if the pattern is at the same orientation, but the different size, it won't cancel out the image. So it means that we have some size selective cells also, not only orientation, but also size. There is an illusion here, like what is this? Which is smaller, the circle, the center circle at the left, or the center circle at the right? Because of the neighborhood, so we might think of the circle at the left as being smaller because of this size selective cells and the size constancy property that we have in our eyes. And yeah, so we can also compare that with the computer vision softwares to that. It seems that it's really important to also look at the size of the objects. So one of the algorithms, techniques that we use in computer vision is the Gaussian Pyramid. we reduce the size of the object and we blur it also in order to get a pyramid of images of different sizes. So we use that to detect objects in different scales also. And yeah, we have some applications here. So multi-scale processing, so detecting objects in different scales, motion analysis like optical flow. I'll talk about optical flow later, but it uses it can use a Gaussian pyramid instead. Also compression. If we have features that can be found in smaller scales or bigger scales, we can use that to compress the image instead, and multi-scale detection. So we can also detect objects if it's really close or far from the camera. Yeah, we saw about edges, but is color important? So can you recognize the number here? At least I can see 74 here. Depending on how you see the colors, you might see different numbers as well. This is a really interesting experiment. And so let's, yeah, which number? Now, can you find any number here? Yeah, this is the same image, but in a grayscale. So it seems that color is also important to recognize objects. And another experiment is, can you see if which is darker, the A and the B? So it seems that the B square is brighter. But if I do that, if I add a rectangle of the same intensity, we can see that that illusion is destroyed. This is because we also decide which color is an object based on the neighborhood, based on, so we have something called color constancy. This is a desired property in computer vision to detect objects regardless of the illumination, because can you here we have an interesting experiment here. So we have the same cube, the same object, but illuminated in red and green. Can you realize that that green square at the bottom is the same as that red square at the top? Do you believe it? I can show it just by converting to grayscale and accept those regions just to see what happens. I will preserve those colors, those regions as well here. So I haven't converted into red the left part. This is the actual color of the square. But because we have a kind of a red illumination, we tend to compensate for that, for the perception of the color. So this is something that is really desirable to detect objects regardless of the illumination. So one example is to try to recolorize the object in order to achieve color constancy. So we can detect objects, yeah, regardless of the illumination here. And yeah, so another example, if we look at this flag for, I don't know, 20 seconds, and here you might think of this, I think you know which flag is this, but the colors are kind of weird because the colors are in an opposite ways, inverted based on the white color. So it means that your photoreceptors are become tired because the image is the same. But if we show now the white. So you might see the appropriate colors for the Swedish flag. Yeah, because the opponent colors are being not fired from the photoreceptors to the brain because they are really tired. Also, we can think about motion. That this illusion happens more on the periphery here. So there is a sense of illusion. It means that our brain also process motion information. And yeah, this is another interesting illusion. It seems like it's being rotated, but this is another illusion. And about motion, here we have two approaches happening in our eye. If we have the eye standing still and the object moving in this way, or if we follow the object, so the object is located at the center of our retina here, but the motion of our eyes could tell our brain that the object is moving. So there's a combination between the changes in our retina and the changes in the movement of the eye here. So we could think of the eye movement as an IMU in our engineering case, where we can track the orientation of the sensor. In the software side, we also have some techniques to detect motion, like optical flow, which is detecting the displacement of the pixels in two images. So we could use optical flow to detect the motion and help us to detect objects as well. Let's see. And one thing which is really interesting is that we don't even need the color information, any and edges, colors, and any kind of information to detect the motion. Let's see if I can play this. I think I can play now. Yeah. So can you recognize the actions, like walking in this case, even though there is no color, there's no edge? So just some features here. So this motivated us to create detectors that captures some features, not only edges, but also key points, like this one. So we could track those features and try to estimate or calculate actions or gestures or the what's happening in the scene. I can also, yeah, this is one experiment that I did, like capturing key points and try to track them in order to maybe detect objects, moving objects. And we can use some descriptors to describe those points: surf, shift, orb, fast, brief, and there are many others also. Also, corner detectors are really important to detect changes in the between different objects. So we could potentially detect key points in that regions. So another behavior in our eye is the motion parallax. We can use that motion parallax in order to see how far the object is from our eyes by looking at the movement, because if it's closer, it moves faster than what is located far from the eye. So we can use that difference to estimate the distance of the object to the eye. And also another interesting information hint or cue that we use is occlusion. So it becomes ambiguous here. This is called the Necker cubes, another experiment. This kind of is ambiguous. Which cube is it? How is it oriented? But if we add occlusion information, it becomes easier here. And so you can see that occlusion gives us information about which is close and which is far, about the understanding the 3D scening. The 3D scene is, yeah. So another aspect is the perspective of the scene. So if the object is far, like twice as far as another object, it will be twice smaller. So we can also use that information to understand the scene also. We can use also texture information, like if we know that the size of those pebbles are about the same, we can also extract information about the actual size in the retina in order to interpret the distance or the perspective of the scene. Yes. Another hint is the use of shadows. Can you see that the animal here is kind of a popping up? It seems that because of the shadow information. But if we look at the animal, yeah, in this second image, we see that it's we interpret as a whole here. So this is because of how the light casts the shadow, and we are used to think of the light casting from above. So like this third image here, I will say here. So if we don't add shadows, so we interpret that ball in a different, in a specific way, but if we add shadow, the interpretation will be really different. So we can use also shadows to try to interpret the scene, what's happening with the scene. So there are some computer vision papers that talk about detecting shadows in order to understand scene. Another aspect here is size constancy. We tend to think the object has having a specific size, regardless of how far or how close they are. So this, yeah, one experiment, which purple line is bigger? Because of the neighborhood, we perceive the purple line as at the right as being bigger, but they are the same. Exactly, and we can see this illusion in this cartoon. So if you see this white line here, it seems to be smaller than this line here, but it's the same size because of the perspective. And there's another illusion called Miss Room illusion here. The room is constructed in a way such a way that the second person can be really far. So it seems that it's really small compared to the person which is close. But the walls are created in order to cancel the perspective sensation or perception of the perspective. And yeah, so there are many applications for this. This inspired us to create a lot of computer vision algorithms, like depth estimation algorithms to compute the distance between the camera and the object, simultaneous localization and mapping, trying to make the robot localize itself at the same time mapping the scene, 3D reconstruction from a bunch of images in different orientations. We could try to estimate the 3D coordinates of those points. And recently, people are looking at social distancing. Yeah, 3D features are really important because the distance between the people, between two persons, are different of the distance between the pixels of both images. So we need to use 3D geometry in order to better estimate the distance between the people. Yeah, and about face recognition. So could you recognize which person it is said? It seems to be easy for us. It's that one. So even if those faces are just a bunch of line drawings, we are really used to detect faces. We can detect faces even on things that does not have a face. And even babies can look more at the face as at the left instead of this possible face at the right. So it means that we are really almost hard coded to detect faces, but it can be also learned. There's another illusion here. So can you see the difference between those images? This is a known face. And you can see that the eyes in that left image are rotated, but it seems kind of reasonable. And the mouth here is also rotated, but the differences become really, yeah, larger or bigger when we see in a correct orientation. This is because we are used to look at eyes and mouth and nose in the correct orientation, but it becomes really difficult when those components become in a different orientation, like that one. It becomes, yeah, you could spend some seconds trying to identify those people, but it becomes really easier when when the people are in a correct orientation. And also intensity also matters. So if we try to see negative images, it becomes a little more difficult to identify faces than to have the correct the correct brightness and the colors. And but if we just show the correct brightness of some specific key point, or like the eyes, we could easily recognize the person. Those are the president, United States presidents. Yes, so let's see. Also, recognizing individuals in a specific group. I don't know if you had this experience before, like living in another country. And the first day you might see the people, that the people seems like almost the same, but as long as you live for 1 year, 2 years, you can see the differences between them. So it seems that face recognition can be also learned somehow in some part. And this is also raises a question about bias in our face recognition algorithms. Because if we supply a data set that is restricted to a specific group, we have to think about bias in our network that might affect the decision for other groups. And that this might be, that might result in bad results, in bad decisions. Also, another aspect is caricatures, which is exaggeration of some aspects of the body. So it makes us to better recognize the person. So we can use that fact to create faces that are less recognizable, like not only exaggerating, but also removing or reducing those features. Like which R node is the correct one, the real one. So in case you say it's the middle one in the middle. This is exaggerated. This is a caricature, and this is it's the opposite. This is a caricature. This at the right, and this is the anti-caricature. It's reducing the features based on this average image here. And but some expressions seems to be more universal, like smiling, crying, and this can be encoded into drawings as well. And we can use that to recognize the actions or the expressions in our algorithms and networks as well. Even cartoons, yeah. And there are some algorithms like deep learning, Viola Jones in the 2000s, eigenfaces that are faces that most represent a distribution of faces in the data set. So we can compare, instead of compare the whole data set, we compare just the face, those eigenfaces. And the histogram of gradients that we discussed before, we can use that to recognize faces as well. And yeah, and deep learning, which has some interesting features, like if we, yeah, deep learning is a set of calculations that we can make in an image, in a video, in audio, text. So to classify, to detect, to extract information from that in a series of computations. So we can extract features in the first layer. So we can extract the features of features. So in a deep representation, so we can try to estimate the high level representation of that image in order to better classify it. So those features in the first layer, if you see, it's mostly like edges, as we saw in the process of the eye, like processing first the edges. If it's a colored image, so we can see we could see also opponent colors as well. And later we can combine them to try to extract more complex features like eyes, mouth, and the later faces, and yeah, really more complex features as well. So we can use that for classifying faces or objects, captioning, image captioning. I will the next slide will show that. Self-driving, reinforcement learning, which makes the computer to play games and learn by itself by optimizing reward function. Image restoration, hallucination, which is creating images from a database. So we can hallucinate, so we can create new images from them. So image captioning, this is one example. So the network can extract or try to extract what happens in a scene as well. Yes. Also style transfer, so you can encode like a painting, we could calculate the features of the painting in order to apply those features to other photos. So we could transfer the style of Van Gogh, for instance. Yeah, and how to learn deep learning in that sense. So those two courses are really interesting to look at it. So I can provide those links here. And also there is a deep learning book, so you can use that to learn deep learning as well. And I created this presentation based on those books, Basic Vision and Sensation and Perception, that to understand more about human vision and how we could be inspired to develop more computer vision techniques. And yeah, that's it. I would like to thank you very much. And if you have any question, so I'm open. Okay, so
Hello,
yeah.
Anderson. It's Michael
Hello.
here.
Yeah.
Thank you very much. It was really, really nice seminar. I would like really to thank you for this information. I have a question regarding the optical flow. Is it
Yeah.
reducing? Can it be, or is it used to reduce the video? Say storlek, which is so you can make a video more compact in some way, or encrypt or decrypt a video. Can that be used?
Yes, yes. So if you calculate the optical flow in a dense way, like you calculated the displacement vector for each pixel, you can use that information to eliminate the pixel in the second image in order to store just the first pixel and the displacement as well. So, and if common region moves uniformly, like a set of pixels that belongs to a region, if they move in the same manner, so you can store just one vector displacement for a bunch of pixels. So you reduce a lot the size of the video as well. So like the sky, for instance, we don't need to store the sky for every frame because mostly they are the same intensity, the same color. So we can just provide the first frame or just the iframes as well. So we can use that also to we can use optical flow to calculate the motion, and based on the motion we can remove parts which are not moving as well, or if they are moving in the same manner, we can just store just one vector. Yeah, it's possible.
Yeah, really good. Thank you for the response.
Yeah. If you have also more questions, you can also contact me at anderson@synteda.se. So I would like to thank you very much.